Good day, everyone. Today we will talk about the techniques in biotechnology. We are now on our lesson 2.3, and today we will focus on DNA cloning and recombinant DNA technology. Here are the topic outlines for this discussion. We will talk about DNA cloning, the restriction endonucleases, the important steps in DNA cloning. We will also talk about the DNA vectors, the cloning vectors, and recombinant DNA technology. Okay, so DNA cloning. When we say cloning, this means we make an exact copy of something. So when we talk about DNA cloning, it's like making an identical copies of our specific DNA. So it's just like we are copying a file on our computer. Basically, that's just the principle about it. To start, DNA cloning is a molecular biology technique that is widely used by scientists if they want to study a specific gene or a specific DNA, or if they want to use it for different experiments. So first, they separate that piece of the DNA or the genes from a larger chromosome. Let's say, for example, from a plant or an animal or a bacteria. So think of it like you're cutting a particular sentence from a long book. So that's how you cut it or how you separate a specific DNA. So once you've got the gene or the DNA piece, you will attach it to a smaller piece of DNA that can carry it, and that is what we call the cloning vector. It's the carrier of the inserted gene. So one prime example or one common example for the cloning vector is the plasmid, which is a small and a circular piece of DNA, and this is often found in bacteria. And to make these cuts and pastes in the DNA, we use what we call the restriction enzymes. So this restriction enzyme can cut and paste the DNA that we want into the vector or into the plasmid with the help of another enzyme for ligation, which is the DNA ligase. After all this, they have a new DNA combination that contains the piece of the DNA that you want to copy or your DNA of interest, which can then be introduced into the bacteria or other cells to make lots of copies. So now this will allow the researchers or the scientists to study it or use it for various purposes, say for example in medicine or in research. So basically it is about cutting out a part of DNA and pasting it into another piece of DNA and then making copies of it. On the previous slide, we talked about DNA cloning as something that we need to make identical copies. So how is it different from the PCR? So here we have a list of the things that we do when we want to do cloning and we want to do PCR. In cloning, we do cloning when you need to store or modify a DNA. So cloning allows the DNA to be stored in living cells, let's say for example in a bacteria, and you can store that for a long time. And these cells can also be grown and they can be multiplied whenever you need more of their DNA. Second is if you want to insert a gene into an organism. So if you want to insert this gene to study its function, or if you want to express it or make a protein, then you should do cloning. For example, if you want to insert a human gene into a bacteria to produce insulin, then you will do cloning instead of PCR. Second is if you want to produce proteins. So if your goal is to express a gene and then you want to produce large quantities of a specific protein, then cloning into an organism is very necessary because the cells will not only copy the DNA but it will also translate it into a protein. Third is when you need a stable and long-term solution. Cloning will allow the scientists and the researchers to maintain a DNA sequence within a plasmid that is stored in the bacteria for other organisms. So the DNA can be grown and they can also be copied indefinitely. Last is when you want to mutate or if you want to modify a DNA. So they use cloning when they want to introduce a specific mutation into a gene or change it into some way before they study it. And this will allow for the manipulation of the gene in every controlled manner. So now, when do we use PCR instead of cloning? So if you only need to amplify your DNA quickly, then you can use PCR. So PCR is perfect when you need to make many copies of a specific DNA segment quickly, which, for example, you need it for sequencing or for diagnosis or for just forensic analysis. Then it can amplify the DNA even from a very small samples. Second is when you don't need to insert the DNA into an organism. So if your goal is to simply get more copies of the DNA for analysis, then PCR is much faster and is more efficient than DNA cloning because it doesn't work or it doesn't involve in working with cells or other vectors. Third is when you have limited DNA starting material. So PCR can start with a very tiny amount of DNA, but still can generate millions of copies. And it's great for forensic science, as I mentioned earlier, or crime investigations, or by just looking at the ancient DNA samples where the amount of DNA is very low. So if you're interested with that, then you can do PCR. Last is when you want to detect the presence of a gene or a DNA fragment. So if you just want to know its presence, then you can go for PCR because PCR is often used for diagnostics. So it can identify the presence of a specific DNA sequence by amplifying it if it's present. DNA cloning is a fundamental technology in genetics, and this technology has made it possible to isolate, to clone, and to produce DNA for all the genes in appropriate quantity so that you can sequence it later on for it to be characterized. So cloning allows us to work with DNA in a more manageable and a more concentrated form. And one of the big advantages of DNA cloning is that it makes it possible to study the genes that are normally expressed at very low levels. So normally we have very low levels of DNA on some parts of our genes, but through this DNA cloning, we will be able to normally express it even in a low level. So in natural conditions, these genes might be very difficult to detect or analyze because they do not produce that large amount of material that it needs to produce. But through this cloning, we can now amplify these genes and we can generate the specific amount of the recombinant proteins that we need for our studies or our further applications. DNA cloning is also the first step in many genetic engineering experiments. So without this, the process in genetic engineering wouldn't be possible. So for example, we want to use DNA cloning to produce DNA libraries, which are the collections of our DNA that represents the entire genome of an organism. So this is useful when we want to identify and if we want to study individual genes within that genome. Another importance for the important application of DNA cloning is the PCR, which we also discussed last meeting. So PCR relies on the DNA cloning techniques because it is used to amplify specific DNA sequence and it is essential for countless applications, say for example in research, in medicine, or in forensics. And also, it is also critical for DNA sequencing because before we can sequence the DNA, same with the PCR, we often need to clone it to ensure that we have enough material that is easy to handle. So by cloning, we get a clear, readable sequence that can then be analyzed and understood in the organism's entire genome. For the process of DNA cloning, first is we isolate the donor DNA gene, and then we select for specific vector, and then we incorporate the donor gene in the chosen vector, Then transformation of recombinant vector to host cell and isolating the recombinant cell. To understand more about the steps in DNA cloning, then we'll discuss this one by one. Before we carry out the operation of gene cloning, we need basic thing in their purified state. First is the gene of interest. So the gene of interest is a fragment of gene whose products actually interest us. So it's the product that is our interest. Might be an enzyme, a protein, or a hormone. For example, a gene that encodes for a hormone insulin, or those gene that encodes for valued traits like, for example, herbicide tolerance or pest resistance, drought tolerance, salt tolerance, or maybe an increased yield. So the target DNA may be genomic DNA or the complementary DNA or other types of DNA. Now, the DNA is extracted from that organism under study, and it is cut into small fragments and that is suitable for cloning. So most often this is achieved by cleaving or cutting the DNA, and uh, an important enzyme will be used during this step, and this enzyme is called the restriction enzymes, particularly or specifically the restriction endonuclease enzymes. So restriction enzymes are also called the restriction endonuclease. They are the ones that recognize a specific sequence of our nucleotide in a double-stranded DNA. We call the restriction site, and they cut the DNA within or adjacent to that site by cutting the phosphodiester bonds between the nucleotides. Again, they cut the phosphodiester bonds between the nucleotides by cleaving. So the restriction enzymes are the ones that will cut the DNA fragments at specific sequences, and the specific DNA sequence is called the restriction site. Again, the restriction endonuclease enzymes, they are the ones that will cut the DNA fragments at specific sequences, while the restriction sites are the specific DNA sequence. That that is where we cut the uh, DNA. So the restriction endonuclease enzymes, they are the ones that recognize that restriction sites. Restriction sites, the area where we cut the DNA, and the enzymes are the ones that cut. So these enzymes will recognize the restriction site or the restriction recognition site of the DNA molecule, and then they will cleave that DNA sequence. The second one is the selection of a specific vector. So when we say vectors or cloning vectors, these are carrier molecules which can actually carry our gene of interest into a host that is responsible for the replication. And these replicate there along with the gene of interest, making multiple copies of it. So cloning vectors are DNA molecules that transfers the foreign DNA molecule into the host cell. Cloning vectors has the ability to also to self-replicate, and they also have the ability to integrate into the host cell. That's why they are used for cloning. The target DNA is inserted into the specific sites of the vector, and they are ligated by the ligase. Again, these target sites or the target DNA or our gene of interest, they are inserted into a specific site of the vectors or our cloning vectors. Like this may be a cloning vector. And then once they are inserted into the vector, they will be ligated or they will be ligated by the DNA ligase. So this vector is then transformed into the host cell for replication. So it is important that we have this cloning vector because it facilitates amplification from a single copy of DNA molecules into many copies. And also molecular gene cloning would also be difficult if we don't have these cloning vectors, because how else would we transfer the genetic material or would we amplify the genetic material without these cloning vectors. So there are actually different kinds of cloning vectors that we can use. And the common one is the plasmids. But there are also other things or other cloning vectors, such as the bacteriophage, the bacterial artificial chromosome or the BACs, yeast artificial chromosomes, mammalian artificial chromosomes. So you can also use that one. But the most common ones that has been used widely in the laboratories are the plasmids. So these are each with different insert size, and it depends on the size of the. So when we use vectors or when selecting a vector, we should also consider the insert size. So what is the size of our gene of interest? What is the size of the vector? What is the size of the restriction? And also the efficiency of the cloning. Also, or I mean, the cloning vectors also affects the efficiency of the cloning. So as I've mentioned, the most widely used vectors are the plasmids, and these are circular DNA pieces, and they are found within the bacterial cells among the bacterial chromosomes. So they are fine to be removed or be isolated because their presence is not that vitally important like the chromosomal DNA on the host. However, the genes that they carry may provide additional characteristics for the host cell and can increase its competitiveness in the environment. So again, these plasmids are from the bacterial cells. So these are removed and isolated from the bacterial cells so that it can carry the gene of interest. So again, the genes, even if it is removed, it is still fine if they are removed into the bacterial host cells because they are not vitally important, but their presence also may provide additional characteristics for the host cell. So an example of a plasmid vector is the TI, or the tumor-inducing plasmid. I think you've already heard about this because this is a very common plasmid, the tumor-inducing plasmid, and this is isolated from the Agrobacterium tumefaciens. So this Agrobacterium tumefaciens has a plasmid which is tumor-inducing plasmid. So this Agrobacterium tumefaciens is a pathogenic bacterium, and this actually causes the crown gall diseases, and this is a plant tumor which affects a wide range of plant species. So mo nang naay mga tumor sa atong kuan mga bukdo-bukdo mga crown galls sa atong kuan sa atong mga plants, especially on the roots. It's because of the presence of this tumor-inducing plasmid nga na insert gikan sa atong bacteria to the plant. Crown galls develop upon transfer of a portion of tumor-inducing plasmid, and they call it the transfer DNA. Again, crown galls will develop upon transfer of a portion of the tumor-inducing plasmid, and this is what we call the transfer DNA. This specific transfer DNA causes the crown gall to develop once they are inserted into the genome of the bacterium's plant host, then the plant host will manifest the tumors in their organ. So tDNA transfer is initiated when the agrobacterium will detect the phenolic molecules that is released from the actively growing cells in the plant wound. So baka once there's a wound in the plant, the agrobacterium will detect it because of the phenolic molecules na na-release sa plant, and mo nang ilang signal for them to uh, initiate the transferring of their tDNA. And these phenolic substances will induce the expression of virulence genes. And these vir genes, kini nga mga gitawag nato nga mga vir genes or the virulence genes, they encode for products that is responsible for processing and transferring the single-stranded tDNA. Again, ang atong tDNA mo to siya ang nakakos sa tumor nga siya ang nakakos sa tumor sa atong plants or tumor formation sa atong plants, and these tDNA are carried or they are transferred by the help of the vir genes. So Sila ang mag-help in carrying this DNA to the uh, to the plant host. So, managitawag sila ang virulence genes. The replication of the plasmid into the host cell is done, it can be done in two ways. It's either by stringent control of replication or by relaxed control of replication. So, mana ganitag stringent control of replication, these plasmids are dependent on the presence of initiation proteins that are synthesized by the host cell para makastart sila og replication. So, it means naapay prerequisite, they're dependent sa presence sa initiation proteins nga gikan sa host cell para makastart sila og replication. Whereas in relaxed control of replication, plasmid can initiate DNA replication independently bisan og wala may initiation proteins nga na-synthesized by the host cell. So they can independently replicate their DNA in the bisag walay host initiation proteins. So they only require the host replication machinery for elongation and termination. And most likely, if we have relaxed control of replication, it tend to be in a high copy number. Unlike atong stringent control of replication, since naman siya prerequisite nga nag-require man siya og initiation proteins ikan sa host cell, then it tends to have low number kay if walay initiation proteins nga available sa atong host cell, so that means ang plasmid is dili pud siya ma-duplicate. Unlike sa atong relaxed control of replication, it does not need the host initiation proteins. It only requires the replication machinery, and it's always there for elongation and termination. So for DNA replication, as long as na yung machinery for DNA replication and terminate for DNA replication, then it can start its replication process. And this results to a high copy number, or mas daghan ang iyang replicate, or mas taas ang number ang kaya niyang replicate. So after no nga natay vector or natay plasmid, and we also have the gene of interest. So now we can incorporate the donor gene in the chosen vector. So once the gene of interest and the cloning vector are ready, we can now start the operation. So again, una gid sa tanan, before man ta mag-start with anything, we need to prepare the materials. And very important materials here in DNA cloning is the gene of interest and the vector. In this case, we will look at the plasmid as our vector. So once the gene of interest and the cloning vectors are ready, now we can start the operation. So our next step will be to cut the both the vectors as well as the gene of interest by using the enzyme sa mag-cut restriction endonuclease. So again, a restriction endonuclease, that is an enzyme that cuts the double-stranded or single-stranded DNA at a specific recognition nucleotide sequence, which we call as the restriction sites. So ang atong gene of interest, we have also restriction sites as well as our plasmids. So they are also regarded as molecular scissors because, again, they cut open the DNA strands. And the same restriction enzyme will also be used to cut or isolate the gene of interest from the cell. Kung unsang gigamit ng enzyme for cutting the plasmid is also used to cut and isolate the gene of interest from the cell. So after it is cut, or the cutting step is done, we move to pasting. So diri after the cut, ibalhin na nga atong gene of interest pasulod dito sa atong plasmid. Here, the gene of interest is taken and pasted to the cut vector. So na-open mo ito ang circular DNA. So dito i-insert ang atong DNA, and then this procedure also needs an enzyme we call the DNA ligase. So siya, di ba, mag-connect sa mga broken strands o if ma-recall na ito atong DNA replication during the Biol 22P.
So, mo gamit gyapon ta og DNA ligase. And this DNA ligase enzyme is considered as a molecular glue. It glues the cut nga mga fragments. So, ang atong fragment from our gene of interest, and then ang atong this one. So, kani siya ang atong gene of interest, and also these are plasmid. So, kani na-cut ni siya by the unsa to, enzyme, by the enzyme, itong enzyme, we have the restriction enzyme. So, this restriction enzyme will mo siyang nag-cut sa atong plasmid as well as our gene of interest. And then once they are cut, it will be pasted. Kaning atong gene of interest will be pasted here. I-insert na siya diri sa atong plasmid or sa atong vector. And once they are inserted, napanay mga gaps, diba? As you can see here in both ends. So this will be glued by the enzyme ligase. So, i-glue na siya because it is a molecular glue ang atong DNA ligase. And after combining the targeted gene of interest and plasmid DNA, the resulting DNA molecule now is a hybrid of two DNA molecules, our GI or our gene of interest and the vector. So, mao siya ang atong recombinant DNA plasmid. We have the genes from the plasmid and we also have the gene of interest. So this gene of interest again may be coming from the genes sa kung sa atong interest, maybe from an animal or a human or a plant, or not a specific sequence lang gusto nato nga studyhan. So in the terminology of genetics, this intermixing of different DNA strands is called recombination. So naa na tay recombinant. This new hybrid DNA molecule is also called a recombinant DNA molecule, and this technology is called the recombinant DNA technology. So from the plasmid, then we insert a gene of interest. So tuha siya ka set of genes that are coming from different nga mga species or different nga mga organisms, and they are fused together. Then they are now called recombinants by the technology recombinant DNA technology. Okay, for the fourth step of gene cloning, so fourth step is the transformation of the recombinant vector to a host cell. We also call it the transformation. So when our recombinant DNA molecule is ready, so nanata atong plasmid with inserted gene of interest. If ready na siya, then we need now to introduce it into a living system, which we know as a host. So usually, we use bacteria as a host. So this is done either for one or both reasons. First reason why we are doing transformation is maybe we want to replicate the recombinant DNA molecule for us to get the multiple copies of our gene of interest. That's the first reason that you would want to, why we would want to do transformation. The second one is we want our gene of interest to get expressed, and then it will produce the protein which is needed by us. Again, first is we want to have a lot of copies of our DNA or of our gene of interest. The second one is we want that gene of interest to be expressed or produce the protein that is needed by us. Or if just maybe we want to know the function of the DNA or the gene of interest, then you can use that one. So introduction of the recombinant DNA into the host cell is done by various ways. And they depend strictly upon the size of the DNA molecule, kung sa kadako, kung sa pila ka base pairs ang naa sa imong plasmid, and kung unsa ang nature sa imong gene of interest. So when we carry out this process, some of the host cells will take up the recombinant DNA and some will not. So not because you are doing the transformation automatically, tanan ng mga bacterial cells o tanan ng mga bacterial cells na gibutang diha will receive the plasmid. So some will take it, but some will not be able to take it. So that's why later on the process, we will do selection to see whether nabatay mga bacterial cells that contains the recombinant DNA or wala. So the host cells which have taken up the recombinant DNA are called the transformed cells. And the process is called the transformation. So, kadto nga mga kuan, kadto nga mga DNA, kadto nga mga bacterial cells or mga hosts na nag-contain na sa recombinant DNA, they are called the transformants and or yes, the transformants, and we call this process a transformation process. So, when we say transformation, it is the process that is carried out to transfer that recombinant DNA molecule into the host cell. Sa ganitong recombinant DNA, those are the plasmids with the gene of interest. So, si gene of interest of the plasmid, they are inserted into a host cell. This is, in a purpose, it's either we want the gene of interest to be expressed, or we want to have multiple copies of our gene of interest. Transformation is also called as competency or the ability of the cell to take up foreign DNA. So, diha po natin mabalaan kung unsa kanindot ang atong host cell. So, we have, like for my experience, or from my experience, we used multiple strains of bacteria because we want to look at the competency or the ability of that specific cell to take up foreign DNA. Different strains may have different competencies. So some may take it, some may take the new or foreign DNA, some may not be able to receive or to take in or to include it in their system. So that's why there's a lot of strains that possess different types or different competencies. So naay daghan nga mga lain-lain nga mga methods that we can follow to carry out these steps. That includes chemical method, electroporation, microinjection, gene gun method, and these pathogen vectors. So I believe this will be discussed also later on as this discussion progresses. Last step is the detection of the recombinant clone and isolating the recombinant cell. So, of course, we want to know if there are already transformed cells or transformed host cells wherein ang atong mga recombinant DNA or the plasmid together with the GI or the gene of interest is already inserted into the host. So, the host cell is allowed to grow in a culture media. However, this might contain both. It's either there's a recombinant and non-recombinant cell because as I mentioned kaganiha, we already carried out the transformation process. And some of the host cells will take up the recombinant and some will not. And of course, it depends also on the ability of the cell kung saan nila pag-take up ang foreign DNA. So the mixture of recombinant and non-recombinant cells needs to be separated for which the marker gene is used. So as we're interested only in the transformed host cells, it becomes necessary kinang nato nga i-filter na sila. So this is exactly what is done in this stage or the selection process. So there are many existing selection strategies, some of which includes taking the help of reporter genes or colony hybridization technique, which is the use of antibiotics. So we have here an example of selection process today of recombinant and non-recombinant DNA or recombinant cells. So remember that plasmids used in cloning should have a selectable marker with an antibiotic resistance genes, and this will facilitate the screening of the recombinant organism. So a plasmid typically contains antibiotic resistance genes, no? So usually, naagit tayo mga antibiotic resistance gene because as I've said ganina during the start, this is very important during the selection process. So this will allow the bacteria to survive in the presence of a specific antibiotic. So during the screening, all of the transformed cells or the host cells are placed on an antibiotic plate to select for ones that took up as plasmid. So we have here colonies, and these bacteria
will be placed on an antibiotic plate. Bacteria without a plasmid will die. Each bacterium with a plasmid will make a colony. So this means na kanhi sila ng mga colony, these are transformed. So those bacteria without a plasmid will die, and the bacteria that carries a plasmid can live and reproduce. And each of the surviving bacterium will give rise to a colony of identical bacteria, and they will all carry the same plasmid. So now it is duplicated. So once we have purified our transform host by screening process, it is now for our job to provide them optimum parameters to grow and multiply. Since na naman tayo colony, then we can now subject them to optimum nga mga conditions for them to grow and para malaghan po sila. So a colony containing the right plasmid is grown in bulk and they are used for plasmid or protein production. So in this step, the transformed host cells are introduced into fresh culture media. So kaning mga colonies, i-transfer na sila dito sa fresh culture media and then it will provide them with rich nourishment and they were followed by incubation at right temperature. So, but you need to note that not all colonies will necessarily contain the right plasmid. That's because during the ligation nato, no, the fragments don't always get pasted. So, maybe na-carry ang plasmid but wala ang gene of interest. So, there's possibility nga na ang itabo. So, instead, we must collect the DNA from several colonies. So that we can see whether each one contains the right plasmid. So, dili lang pwede nga usa ra ka colony kanilang usa lang ka colony atong kahaunaan i-reject na nila atong uban because there's some instances nga ang plasmid day ang naa pero wala tong atong gene of interest because diba ang plasmid nag-carry og resistance genes. So if na-insert ang plasmid pero walay resistance gene, na-insert ang plasmid walay gene of interest, it still doesn't make sense. Wala na sense no ang atong ginabuhat because we are actually interested in gene of interest. So, kuha na to, kung sa mga nabuhi, ato na siyang ipang-streak or padaghanon nato na siya in the right video nga kanang optimum for them to grow and flourish. So, methods like enzyme digestion and PCR are commonly used to check the plasmids. So, i-check na po na nila nato kontra. Kung sa man kataas sa katong i-expect. So, anong kinanglan maday natong i-check? So, ang bacteria that makes colonies should all contain the plasmid. Diba? Kung kinsa to mga nakasurvive ang nag-create of colony, they should contain the plasmid because this plasmid provides antibiotic resistance. However, it is not necessarily the case that all the plasmid that contains colonies will have the same plasmid. How does that work? When we cut and paste the DNA, it is often possible na naay uban nga mga products na nahimo or na-build in addition to the plasmid that we intend to build. For instance, when we try to insert a gene into a plasmid with the use of a particular restriction enzyme, basin natay in some cases we get some cases where there are plasmid that closes back up. So bisan og wala nila nakuha ang gene of interest, bisan og nagkuana na og restriction enzyme ma-cut na siya but there's some instances na mabalik na po na siya og close. So because of that dili niya ma-take in ang gene. And other cases also na ay mga genes nga mo atras or mohawa og balik before pa man nga ma-ligate siya sa DNA ligase. Okay, why are the DNA vectors important? So the DNA vectors or these cloning vectors are the carrier molecule which actually can carry the gene of interest into a host and then they replicate there along with the gene of interest which makes multiple copies of the DNA. So cloning vectors are DNA molecules that helps in the transferring of the foreign DNA molecule into the host cells, and they are like a vehicle or a transporting vehicle um, or a vehicle for transporting this foreign genetic material into another cell. And of course, the foreign segment of the DNA is replicated and then they are expressed using a machinery of the host organisms. So the agrobacterium transformation method relies on the soil bacterium, which we name the agrobacterium tumefaciens. This is to transfer the gene of interest into the genome of the plant cell. So agrobacterium tumefaciens, as you know, is a soil-borne, they are rod-shaped, motile, and bacterium, and it is a causative agent of a very common disease, the crown gall or the hairy root disease in many plants, such as the dicotyledonous plants, in which the tumor-like tissues grow in the roots of infected plants. As you can see, ani nga picture kani siya. So this agrobacterium is attracted to the wounded sites of the plant's roots via chemotaxis, and that is in response to the release of the chemicals such as sugars and phenolic components of wounded sites. So if the plant is wounded, it gives signal to the agrobacterium to infect it because they are highly attracted to phenolic compounds. So in wounded sites, they release phenolic compounds, and that is when the agrobacterium or the bacteria will infect the plants, and the agrobacterium will also affect the plant there and will cause the gall formation. Now, it is found out by several scientists in the past that agrobacterium is able to transfer its bacterial genes into the plant genome naturally, and thus it induces the plant to produce more auxins or more hormones, more auxins and cytokinins than it should, which causes the plant cell to proliferate and they form galls. So this is basically because of the hormones that is produced from the uh, from the Agrobacterium tumefaciens. So this event plays the central role in this gene transfer, and this bacterial plasmid, which we call the tumor-inducing plasmid, is the one that contains the tDNA or the transfer DNA region, which carries the genes and that encodes for proteins that is involved in the production of plant growth hormones such as auxins and cytokinins. And this will cause the plant cell to proliferate and form galls. So, mo ni siya ang mga galls. And this is the plasmid with the tDNA region. So the tDNA region is what is inserted into the host cell. Crown gall formation will occur whenever the bacterium releases the Ti plasmid into the plant cell cytoplasm. And a fragment of Ti plasmid, this one, the tDNA, is actually transferred from the bacterium into the host where it gets integrated into the plant cell chromosomes. So, mao lang ni siya ang ma-integrate dito, and this is by the help of the vir genes or the virulence genes. So, virulence genes aids in the transfer of the tDNA from the plasmid to the host cells. So, we can um, we can now manipulate this natural process and we can replace this tDNA with our desired genes. So, pwede natong tanggalon from the plasmid, tanggalon nato ang tDNA, which is the region where the region of the plasmid where it is inserted to the host cell. Pwede natong alisdan ang tDNA to a desired trait or to desired gene. Then the agrobacterium will now be able to transfer the desired gene into the genome of a target plant. So, i-remove ning natural nila nga tDNA region, replace with our gene of interest, and then agrobacterium na ang bahala nga mo transfer sa atong gene of interest to the host plant or the host cell. That is by the help also of our regions or their genes. Okay, we've been talking a lot about Ti plasmid, but what is this agrobacterium Ti plasmid? So the central role in this gene transfer is the bacterial plasmid, that is what we call the Ti plasmid. So this gall formation, crown gall formation, occurs when the bacterium releases its Ti plasmid into the host plant cytoplasm. So the Ti plasmid from different strains of Agrobacterium tumefaciens has some features in common. One of those features is the regions of the tDNA or the transfer DNA. So this tDNA or the transfer DNA is a fragment of the Ti plasmid. So salo na siya part sa plasmid where it actually is the one that will be transferred from the bacterium into the host where it gets integrated into the plant cell chromosomes. Say for example, in sa atong plasmid, we only need this tDNA nga mo insert dito sa atong plant para maka-cause siya og tumor. Next is the Vir regions or the virulence regions or the Vir genes. So this is another important component sa atong Ti plasmid. This component. This is very important because this Vir regions is responsible for the transfer of our tDNA into the plant genome. So the activity of the Vir proteins will promote the tDNA integration into the plant genome. And there are actually at least about nine virgins or virgin operons that are identified in Ti plasmids. And they are the ones who have the tDNA to be transferred from the plasmid to the host cells. Another important part of the plasmid is the origin of replication. So it is the sequence of nucleotides where the replication initiates. And this should always be present for autonomous replication sa atong mga Ti plasmid inside the host cells. And then we also have here a region enabling conjugative transfer. And this has a restriction site for the insertion of the target DNA. And lastly, here we have the genes for catabolism of opines. So here, it also has opine synthase genes and it encodes for enzymes na involved for opine synthesis. So, important ni siya because it carries genes which encodes for proteins that is involved in the production of plant growth hormones as well as in the biosynthesis of novel metabolites. Here we have four important features of a cloning vector. First, it should have an origin of replication. So it is a specific set or sequence atong nucleotides where the replication initiates. So this is very important, especially because we want to have more copies of our gene of interest. So the cloning vector must have replication, origin of replication. Next is the cloning site. It is the point of entry or analysis for genetic engineering. So we also have here the vector DNA and at this site, it is digested and the foreign DNA is inserted with the aid of restriction enzymes. So diri na to insert ang atong So sa atong cloning site tira na to i-insert ang atong gene of interest. Kay mao ni siya nga region also or section also nga mao ni nga section ang i-insert dito sa atong host cells. Third one is the selectable marker. So this is important, especially if you want to select for the vectors or the host nga na ay successful integration of our recombinant DNA. Then we need to have selectable markers. Most of the selectable markers are katong na antibiotic resistance. So if ang atong host cell kay resistant to antibiotic na, then there's more likely nga siya nag-carry na siya sa atong cloning vectors. I also have marker or reporter gene. It permits the screening of successful clones or recombinant cells. This is very particular or this is very important also in gene expression. So there are general characteristics sa cloning vectors na to na kailangan natin i-consider when we want to select a properly suitable vector for transformation process. First, it should be small in size so that it can be easily integrated into the host cell. Okay, the larger the size, the more it will be difficult for it to integrate into the host cell. So as much as possible, we want to select a cloning vector that is small in size. Second is a cloning vector should possess an origin of replication so that it can self-replicate inside the host cell. So without origin of replication, how will they replicate? So it is very important that the cloning vector should have an origin of replication. Remember that the origin of replication is a specific set or sequence of atom nucleotides where the replication starts or initiates, and they should be considered especially for autonomous replication of the cloning vector inside our cell. Next is it must be compatible with the host organism, of course, no? So there are mga host cells that cannot accept other cloning vectors. So na sila yung mga specific na mga cloning vectors na ilang kinahanglan. So kinahanglan na nga nato kung mabalaan kung unsa nga mga specific cloning vectors ang suitable for a specific host cell or host organism. Fourth is it should have a restriction DNA site for the insertion of the target DNA. So, mo na restriction site, mo na siya ang specific site at which the restriction enzyme will cleave the DNA or cut the DNA so that we can insert our target DNA. Next is that the introduction of donor fragments must not intervene with self-replicating properties atom cloning vector. And then it should have also a selectable marker with an antibiotic resistance gene, and that will facilitate screening of the recombinant organism. So this selectable marker will confer the resistance to particular antibiotic or selective agent, which if in normal condition, it is fatal for the host organisms. But if it is already successfully, if the recombinant DNA is successfully introduced to the host organism, then mabuhay sila. Next is it will confer the host cell the property to survive and propagate in culture medium containing the particular antibiotics, and this is very important ang kaning atong selectable markers because it facilitates screening of the recombinant organisms. Number 7 is it should be capable of working under the prokaryotic and eukaryotic systems. So there are cloning vectors that only works on prokaryotic organisms, but if they are subjected in a eukaryotic organisms, dili na sila mo work. Last is it should possess multiple cloning sites. Cloning site is the point of entry in which the vector DNA at this site is digested and the foreign DNA is inserted with the aid of destruction enzymes. So, kasagaran natin mga cloning vectors, they arbor up to 20 restriction sites. So, they should have multiple cloning sites. Multiple cloning sites are also a feature that allows the insertion of our foreign DNA without disrupting the rest of the plasmids. That's the good thing with the multiple cloning sites.